Hello everyone. Today we will discuss about the DNA replication, and we have divided the part into different um, part of DNA replication. And part one will be we will discuss about prokaryotic DNA replication, and it will be the overview of prokaryotic DNA replication in part one. And in part two, we will discuss the mechanism in different step of prokaryotic DNA replication, that is in the initiation, elongation, and termination phase. And in module 3, we will discuss about eukaryotic DNA replication. And in 4, we will discuss about the DNA damage and repair mechanism. So, hello everyone. I'm Dr. Bandana Padhan from Department of Biotechnology, School of Life Science and Biotechnology at Amas University. Let's start discussing about the overview of DNA replication. Usually, as we know, the part of central dogma, which is a process to translate genetic information into the protein, where the genetic material is stored in the form of DNA and RNA within a biological system. And this information is translated into protein through a process called central dogma. Genetic information from DNA is transcribed to RNA and then translated to protein that are functional biomolecules in the biological process. And in prokaryotic DNA replication, the DNA replication usually happens in the cytoplasm of the bacteria or the prokaryotes, because prokaryotes don't have any true nucleus, whereas eukaryotes have the true nucleus. So this prokaryotic DNA replication usually happens within the cytoplasm of the bacterium. As we know, the DNA replication, which is a part of central dogma, is nothing but the copying the genetic information from for transmission to the next generation. And usually in eukaryotes, it occurs in the S phase of the cell cycle. And we can say it is a DNA duplicating process also. In the overall idea is about, as we know, DNA is a double helical structure. And during DNA replication, it will start unwind the double helix to expose the bases, individual bases of each strand, where the DNA is synthesized by making a new copy of new strand of DNA by taking a parental strand as a template. In each unpaired nucleotide that will attract a complementary nucleotide from the medium or the system or the within the prokaryotes. And for this, various enzymes are required that will link the nucleotide, the additional nucleotide, and they linked the nucleotides by phosphodiester bond to form a continuous strand. This is all about the DNA replication process. And this DNA replication process was proven to be a semi-conservative one. What is that semi-conservative model of DNA replication? Semi-conservative means out of two strand, means the two strand of DNA, individual parental strand will be act as a template for making new strand. And within that model, there are many models were proposed previously, but semi-conservative model was proven. And those model are semi-conservative DNA replication model, conservative DNA replication model, and another one is dispersive DNA replication model. In conservative type of DNA replication, parental strand will be conserved and there will be a synthesis of new strand. So there will be no exchange of genetic information from one generation to another generation in case of conservative type of DNA replication. But in case of semi-conservative, as we discussed, semi-conservative model of DNA replication means individual strand of DNA will act as a template for making a newly synthesized DNA. So in semi-conservative type of DNA replication, there will be exchange of genetic information from one generation to another generation. And in case of dispersive model of DNA replication, what happened? Here it was proposed that the segment of parental DNA strand will act as a template for the newly synthesized strand. But later on, Michelson and Stack, they did one experiment for proving that semi-conservative model of DNA replication is usually happen in both prokaryotes and eukaryotes. So in this experiment, he was the team was culture E. coli cell in a medium where the medium contained ammonium chloride and NH4Cl, and that ammonium, the nitrogen was labeled with N15 isotope. The nitrogen was labeled with N15 isotope. So when the E. coli was grown in a medium containing heavy isotope, then all the nitrogenous bases of the E. coli DNA that will be incorporated by the heavy isotope. And when they isolate the DNA from cell, they found that all the genetic information or all the DNA, they gave a heavy band in a cesium chloride density gradient centrifugation. So they the heavy band was due to the presence of heavy isotope N15 isotope in the nitrogenous base of the DNA. So this was the first generation experiment, and previously they have cultured the E. coli, and it was a control system. Control means they didn't apply any isotope there. So this control system having only the nutrient media containing the light isotope or the general isotope N14 medium, and they extracted the DNA from the cell, and thereby within the after the centrifugation, what they found that a lighter band, because the nitrogen is containing the N14 and the band is lighter. So when they cultured the E. coli in heavy medium or N15 medium, the band was heavy band. And after second generation, what they did, that same E. coli, they just culture or replicate the E. coli in a medium containing again a light isotope or N14 isotope for at least they culture it for 30 minutes, the second generation, and they centrifuge the they extracted the DNA, centrifuged it in cesium chloride density gradient centrifugation, and they found that there was a medium band, means that band is an intermediate band between the light band and heavy band, and that band may contain, they predict that that band may contain N15 and N14 nitrogen with the DNA. So this is the second generation where they found a medium or intermediate band. And the third generation, what they did, again, the system was continue. That means the cell were again continue for the rest 30 minutes within same medium and 14 medium. So when they extracted the DNA after the third generation, what they found that the DNA, the extracted DNA from the cell that contain both hybrid band or the intermediate band and the lighter band. That means the parental DNA is transcribed when during the replication, the parental DNA get duplicated and they make a newly synthesized DNA. So that's why they found equal proportion of hybrid bands and all lighter band. So within for this result, they confirm that the prediction of semi-conservative model of DNA replication was confirmed by this experiment. Okay. So if we analyze the system with the lighter band and heavy band experiment, then semi-conservative model of experiment that gives a lighter band and a intermediate band. The lighter band contain N14 isotope, whereas the intermediate band contain N14 as well as N15 band. But if it was conservative type of DNA replication, then the scientists may got only lighter band and the heavy band in the third generation. And if it is a dispersive type of replication, then they only found a hybrid band. They didn't find or the chances of getting heavy and lighter band is very low or insignificant. Rather than they got or they may get a hybrid band. So this is the prediction of the models, and later on it was proposed that the DNA replication for eukaryotes and prokaryotes are always semi-conservative in nature. So let's discuss about the mechanism or the overall enzyme, overall protein used in DNA replication process. As DNA replication is always semi-conservative, it is bidirectional in case of prokaryotes, and it is semi-continuous. Why it is semi-continuous? As we know, DNA replication is always continue, or there will be a synthesis of continuous strand and a discontinuous strand. That's why it is semi-continuous in nature. So in this mechanism of DNA replication, there are various enzyme and proteins are involved in case of prokaryotic system. What are those enzymes? The main key enzyme is your DNA polymerase. And these DNA polymerase in case of prokaryote are of three types. One is DNA polymerase 1, DNA polymerase 3, and DNA polymerase 2. So this DNA polymerase out of three types of DNA polymerase, only one is main key enzyme for the synthesis of DNA, that is your DNA polymerase 3. Whereas rest of the enzyme, that is DNA polymerase 1 and 2, that are used for the repairing purpose and for making filling the gaps between the Okazaki fragment during elongation process. And additional enzymes like your DNA helicase that usually unwinds the DNA, that means that help in melting the double stranded of DNA and formation of replication bubble or replication fork. Another protein, that is single stranded DNA binding protein, is required for keeping or preventing the double stranded, preventing the rewinding of the double stranded DNA. So as we know, DNA have the capacity of reannihilation. Once we separate the strand, they have the capacity to rewind again. So that protein mainly useful for preventing both the strand for rewinding. And another enzyme, that is DNA primase, or these primase mainly useful for giving or adding RNA primer to initiate the strand in both lagging and leading strand. Uh, why RNA primer addition is required? Because DNA polymerase 3 itself can't synthesize the DNA until unless it will not get a reference. So these primase mainly add the RNA primer or the segments of RNA to start the replication by the DNA polymerase 3. Ligase, another enzyme, is required for making or to seal or weld together the Okazaki fragment or the fragmented DNA into a single DNA strand. So, and some bunch of proteins are required during the DNA replications. These proteins are DNA A protein. The molecular weight of DNA A is 50,000 and it has only one subunit. The function is it recognizes the origin of replication sequence in case of prokaryotes. So in case of prokaryotic DNA replication, there is the DNA is always a circular DNA and it has only one originum site. So this origin will be recognized by DNA A protein. After that, DNA B, which is also known as the DNA helicase, has 300,000 means 3 lakhs molecular weight, and it has 6 subunits. It has 6 subunit of hexameric in nature. So it mainly unwinds or open the double strand of DNA. Whereas another protein, it helps the helicase activity for binding the DNA B to the DNA strand. Another protein is your DNA C. And the molecular weight is 29,000 and it has only one subunit and it assists DNA B to bind to the origin of replication site and also synthesize the new strand of DNA. And DNA polymerase, as we already have discussed, DNA polymerase are of 3 types, 1, 2, and 3. They usually synthesize the new strand of DNA. Another protein, DNA G protein, or we can say it is a primase enzyme, which the molecular weight is 60,000 and it has only one subunit and it synthesizes the RNA segments or RNA primer. The protein, single standard DNA binding proteins, the molecular weight is about 75,000 molecular weight and it has 4 subunits and it binds the single strand of DNA. Another enzyme is your DNA gyrase or DNA topoisomerase. DNA topoisomerase are of 2 types, one DNA topoisomerase 1 and topoisomerase 2. And these topoisomerase usually relaxing enzyme. That means when the helicase activity will be there, so there will be a supercoiling of DNA in the downstream and upstream region of the DNA. So this supercoiling will be relaxed or relieved, the strain will be relieved by the topoisomerase 1 and topoisomerase 2. So the key enzyme, that is DNA polymerase, which is a DNA-dependent DNA polymerase, and it synthesizes the DNA from additional TNTPs. And it requires a template strand where this template is always given by the primase enzyme, and these primase enzyme add primer to the strand having 3-OHN, and all synthesized from 5' to 3'. And if we compare the DNA polymerase 1 and DNA polymerase 3, then DNA polymerase 1 have 5' to 3' exonuclease activity. That means it has a repair activity. And repair exonuclease 5' to 3' exonuclease activity means they can remove the mismatch bases up to 10 nucleotide, more than 10 nucleotide. Whereas 3' to 5' exonuclease activity means they also can remove the single nucleotide mismatch from the sequence. It is a these exonuclease activity have the proofreading ability also. The processivity of DNA polymerase 1 is low, whereas they can add nucleotide per minute is about 600 nucleotide per minute they can add, whereas they only have one subunits. Whereas DNA polymerase 3 don't have 5' to 3' exonuclease activity, whereas they have high processivity and high proofreading ability, as well as 3' to 5' exonuclease activity they have, and they can add 1000 nucleotide per minute and within the sequence, and they have 10 subunits, and individual subunits have different function. These DNA polymerase 1 or Pol1 usually it was discovered in late 50s and discovered by Arthur Kornberg, with who got the Nobel Prize for the physiology and medicine. And these DNA polymerase 1 have 5' to 3' polymerase activity, 3' to 5' exonuclease activity, 5' to 3' exonuclease activity, and it is a high molecular weight enzyme. The molecular weight is about 109 kilo Dalton, whereas if the fragment is proteolized, then it will be fragmented into a smaller fragment and a larger fragment. The larger fragment is about 75 kilodalton in molecular weight, whereas the smaller fragment is about 34 kilodalton in molecular weight. And this polymerase 1, this fragment of polymerase 1, the smaller fragment have the 5' to 3' activity, exonuclease activity, and the larger fragment it is also known as clino fragment also. So these larger fragment have the ability to polymerization, and it has 3' to 5' activity. What does it mean by proofreading? Proofreading means if during the DNA replication any wrong nucleotide will be added, then due to this 3' to 5' activity, that mismatch nucleotide will be removed, and the synthesis will be continued by the DNA polymerase. As we already know that DNA polymerase 3 have 10 subunits. These subunits are alpha subunit, they have epsilon, they have delta. These three subunits are the core catalytic subunit of the polymerase 3. And these three generally help for polymerization activity and for 3' to 5' proofreading activity. Whereas tau subunits, individual DNA polymerase 3 have 2 tau subunits, and usually this tau subunits is for stable template binding and for core enzyme dimerization. Whereas it has also a clamp loader subunit and a clamp, beta clamp we can say. So the clamp loader are gamma, delta, delta prime, then chi and phi. So these gamma, delta, delta prime, chi, phi, these are the clamp loader subunit of polymerase 3, whereas beta are the clamp, DNA clamp that requires for the optimal processivity, and this usually beta clamp is required for dragging the DNA polymerase towards the new template where strand will be synthesized. So, and usually chi is generally useful for interacting with the single-stranded DNA and recognize the site for binding of DNA polymerase and start the DNA replication. Whereas 5 is usually helpful for interaction with the gamma and chi subunit. So here the core subunit are alpha, epsilon, and theta. Sliding clamp are two beta subunit. Clamp loader are gamma, delta, delta prime, chi, phi, and tau. So as we already have discussed, DNA ligase, the main function is to join the Okazaki fragment, and helicase, usually the DNA B protein is a hexameric protein and it unwinds the DNA. Single-stranded DNA binding protein prevent the strand from re-annealing and from being degraded. Gyrase, topoisomerase 2 usually relax the supercoiling of DNA. So thank you all. This is all about the overall prokaryotic DNA replication. So in this video, we have discussed the semi-conservative model of DNA replication, the experiment of semi-conservative model, and different enzymes and protein involved in the DNA replication process. Thank you so much. In next video, we will discuss about the initiation, elongation, and termination process of prokaryotes.